feel like an Amy girl. Okay. Thanks very much, Phil, and thank you for inviting me to speak to you today. I guess now I'm feeling like a Christmas tree. Okay. How's that? That better? Good.
They ran you back and I'll keep it behind you.
Okay. Great, thank you. So while we're getting hooked up, I guess relative to Helen, I am the new kid on the block. I've been working at this for about 20 years now. But I think the work that Helen's been doing on the clinical side has complemented the work that we've been doing really nicely. We've been on the other side of the continent focused on the genetics and the biology of Rett syndrome. And so I was asked to give a presentation on research into Rett syndrome with a particular focus on therapies that are evolving. So just to reorient you, so as Helen said, the main player in Rett syndrome is a gene called MECP2, or methyl CpG binding protein 2, and it's recognised as being a regulator of other genes. So it controls other genes being, we initially thought switching them off, but now we know that MECP2 can actually switch genes on as well. It depends which gene it is. And so that suggests to us that MECP2, the protein, is going to have specific targets that it acts on, and potentially those targets could be where we could be going with therapies. So when we think about therapies for Rett syndrome, right now the mainstay of treatment, which is very effective in quality of life and improving survival and general overall health, is a multidisciplinary approach to management of the various issues associated with Rett syndrome. And this is a list here of the kind of things that we see. And I'll just put in a plug for our clinic in Sydney. That was established over 15 years ago now, and currently run by Carolyn Ellaway. And it was the first multidisciplinary Rett clinic in the country, and one of only a few at the time around the world. So Carolyn has developed a wealth of expertise over the years and has got a really good team working with her. But I think, just to temper everything, right now there's no proven specific curative therapy for Rett syndrome, but that's what we're working towards. And when you think about what kind of therapies you might want to throw at Rett syndrome, it depends. You can think of different components or different approaches. So at one level, we can think about symptomatic treatment, which is the multidisciplinary clinic. At another level, what about if we replace the defective MECP2 gene, so something like gene therapy? What about the downstream targets? As I mentioned, could there be drugs that could be applied to specific downstream targets? And then other factors as well relating to what we understand about the biology of the disorder. So when it comes to trying to unravel the biology of disorders like Rett syndrome, in the laboratory there are a number of different approaches that we need to use. One is the use of cell culture systems, so we can grow cells in a Petri dish, and then we can do all kinds of studies on those cells that can help us to unravel some of the complex biology there. And there are new technologies that have become available over the last 5 years or so that allow us not to just look at one gene at a time, but to look at hundreds or thousands of genes at a time and look at the subtle differences in terms of which ones are switched on, which ones are switched off, and that might help give us profiles with regards to how to tackle the disease. And then finally, you can do stuff in the test tube, but ultimately it's what's going on in real life that's important. And so along the way, a number of animal models, predominantly mouse models of Rett syndrome, have been developed. So these are mice that have mistakes in the MECP2 gene, and which demonstrate various features that we can recognise as being what we see in girls with Rett syndrome. And so you can have all kinds of fun making knockout mice. So a knockout mouse is one where the gene's literally being knocked out, and of course you can then do get mice to do all kinds of tricks for you. to allow you to study the effect of any potential therapies that you might want to be instituting. And one of those things that we did in collaboration with colleagues in Melbourne was to look at this concept of environmental enrichment in Rett syndrome. So essentially environmental enrichment is about providing a really stimulating environment for the mouse, and so and then looking at motor and behavioural abilities of the mouse. And so this just shows you what we mean, at least in the mouse system. You can see the cage on your right has lots of interesting, colourful objects for the mice to play in. And this kind of approach has been used to study whether exposure to an enriched environment can improve overall behaviour. And you can see here there are a number of animal models for human diseases where that's been done, and including in the Rett syndrome. So this is one of the things we do. It's called a rotor rod, where the mice are asked to balance on a spinning wheel, and that's one of the things that goes awry in the Rett syndrome mice. And so Marie Kondo, who was a PhD student in Tony Hannon's lab in the Florey Institute in Melbourne, used one of our mouse models to study environmental enrichment. And just to walk you through this graph, normally when a mouse is exposed to a rotor rod, there's a period of learning, so the ability to stay on the rotor rod improves and stabilises with time. In the female mice with Rett syndrome mutations, firstly you can notice that they can spend less time initially on the rotor rod. They do have an improvement, but then they lose that improvement. In the female mice, when they're exposed to an enriched environment, so that's these mice here, you can see it goes up, and it's very much like what you see in the normal mice. So that provided evidence that environmental enrichment is beneficial in the mouse. And similarly, that also happened at the level of the brain in terms of chemicals. And so an important chemical or protein in Rett syndrome is this one called BDNF, brain-derived neurotrophic factor, and you'll hear that mentioned again. And it's reduced in the brains of the mice, and with environmental enrichment, it started to come up. It didn't reach significance, but it did trend up in the right direction. And then more recently, and this has just been published or just about to be published, Marie also showed that environmental enrichment improved anxiety-like behaviours in the mouse, depressive behaviours in mouse. You wouldn't have thought you could tell if a mouse was depressed, but there are tests to do that. And also one of the stress hormones is quite high in the mice, and environmental enrichment led to this stress hormone coming back down towards almost normal. So all of these things lead us to substantiate what people have observed, and that is that an enriched environment, physiotherapy, music therapy, hippotherapy, etc., all do have a basis to being beneficial in girls with Rett syndrome. So that's just an example of how animal models can help us to build a case for certain therapies. So what about therapies at the level of the MECP2 gene? So I guess the thing that turned everything on its head was the work of Adrian Bird and his colleagues in Edinburgh that was published in 2007. And prior to that, we had all thought, or many of us had thought, that once the neurological manifestations of the disease were there, they were permanent and it was irreversible. But what Adrian was able to do was a very neat trick where he took mice with MECP2 mutations that were already very symptomatic and was able to switch on a working copy of the gene. A very artificial situation, you can't do it in patients yet, but was able to switch on a working copy of the gene, and he reversed the phenotype. So these were mice that were really very sick, not very mobile at all, and I'll show you a video in a second to show that. So he did that by switching the gene on in brain cells, the neuronal cells. And that was an amazing turn of events. It just staggered everybody. And then a few years later, a group in the US did exactly the same thing, but this time in a different type of cell in brain called astrocytes, which are the support cells around the brain. And then most recently, a third group reported work on this time just doing the same thing by replacing microglial cells, and they're like the scavenger cells in the brain, by doing bone marrow transplant in a mouse model. Now, I need to hasten to say that there are at least 2 other groups now who've tried to reproduce those studies and haven't been able to. So I think the jury is still out on microglial cells. So bone marrow transplantation is off the books at the moment as a potential option. And this is just a cartoon showing you the different cell types. The neurone is the light blue cell. That's the main player in terms of communication from one brain cell to the next and sending messages through and so on. The astrocyte in the bottom corner there, that's the support cell that helps neurons do their job and are also important for a whole lot of other functions we're recognizing. And then the final cell is the microglial cell, which is the scavenger cell that is involved in clearing all the rubbish. So this is a video of Adrian's work. So this is a male mouse at about 13 or 14 weeks, and they don't survive much beyond that. And you can see this mouse is not doing the normal thing that a mouse does. It's sitting there, not moving. It's actually trembling, and it's got breathing abnormalities as well. And then on the other side is the same mouse. And I should just point out that behaviour there with the back hind limbs coming together is the mouse equivalent of the hand wringing that we see in girls with Rett syndrome. Then the second mouse is the same mouse treated where the gene was switched on over a period of about 4 or 5 weeks, and you can now see this mouse is behaving much more normally. It's inquisitive, walking around, interested in its environment, and in a second the scientists will pick up the mouse and you'll see the hind limbs are now splayed out, and that's what mice normally do. So that was really, when Adrian showed this at one of the Rett meetings in 2006, it just blew everybody away. And so that reinvigorated us all to start looking for potential therapies for Rett syndrome. So that leads people to ask, well, what about just switching on the normal copy of the MECP2 gene? Girls have 2 copies. One's normally switched off, the other one's normally switched on. So how about we switch on the one that doesn't have a mistake in it? There are problems potentially with that. We know that twice as much MECP2 as is normally present is not good, it's toxic. And there are individuals who have so-called MECP2 duplication, so they have extra copies of the MECP2 gene, and those individuals have intellectual disability and other neurological problems, different to what we see in Rett syndrome, but still significant. And there's also been a mouse model made, I think Huda in fact made the first duplication mouse model, which again showed that the mice, if they have an extra copy of MECP2, have significant neurological problems. There's been additional work in the brain which tends to suggest that you probably only need to have a little bit of increased MECP2 to have an improvement in activity, in neurological function. And some unpublished work by a guy called Steve Gray in North Carolina using gene therapy showed that even by increasing the amount of MECP2 in the gene in the brain by a few percent led to significant improvements in survival. And he's been working on looking at whether that also translates to improvement in various motor and behaviour abilities as well. So all this is to say that if people are going to be tinkering around with increasing or activating the normal version of the MECP2 gene, it's going to need to be very tightly regulated, because too much is going to be toxic. And so that's going to be a challenge, I think. What about repairing the damaged MECP2 gene? One potential approach relates to one particular type of or class of mutations. There are one class of mutations called gene mistakes called nonsense mutations, and what they do is they lead to the machinery within the cell stopping to make the MECP2 protein prematurely. So you get a shortened protein, and that may be unstable and fall apart. And there are a class of drugs that can trick the cell machinery to read through that mistake and make a more or less normal version of the MECP2 protein, which then would hopefully be functional. And so along the way, several groups have made mice models that have specific nonsense mutations. So the R168X is one of the more common Rett mutations that we see, and so Peter Hupke in Germany and his group made this mouse and then have been trialling a number of so-called read-through agents. And then in addition, Caroline Shannon and Jeff Newell in the US were working on the R255X, which is another relatively common MECP2 mutation in humans. I haven't seen Caroline for a few years. I'm not sure if she's still working in Rett syndrome, and Kathy may know, and I can have a chat with her later. But so this is work that could be potentially useful for up to a third of individuals with Rett syndrome. And this is this so-called personalised medicine or precision medicine that President Obama talked about last year. So I mentioned that there are targets. MECP2 switches on some genes, switches off some genes. So these could be potential targets for treatment for Rett syndrome. And the trick, of course, is identifying which is the right target to be going for. Don't do this at home, folks. And so along the way, there have been a number of already published clinical trials working on putative targets of MECP2. Helen mentioned the carnitine study that Caroline did as part of her PhD studies about 20 years ago. There's been work published on the ketogenic diet, so it's a high-fat, low-carbohydrate diet to improve seizures, and certainly in some girls with Rett syndrome that works. The question is whether that's got anything to do with the biology of Rett syndrome or whether it's just a non-specific thing, because the ketogenic diet is used in other seizure disorders and can have similar kinds of results. MECP2, the first part of the gene is called methyl, and so there was at one stage there was a thought that if you could increase the amount of methyl groups on DNA, that might allow residual MECP2 protein to be able to do its job more effectively. And so treatments with various so-called methyl donors as oral therapies were tried, which showed no real evidence of improvement. So that's sort of been scratched off the list. And then naltrexone, which is an anti-opiate drug, has been used and showed some improvement in the respiratory disturbance in some patients with Rett syndrome, but it didn't seem to affect the overall severity of the disease. There's also been work done in mouse studies, so I won't go through all of these, but I'll come back and talk about histone deacetylase 6 inhibitors, because that's some work that we've been doing more recently. And then there's some unpublished work on several drugs that could be potentially useful. The first one is work being done by John Bissonnette in Portland, Oregon, and which appears to improve the apneic episodes associated with Rett syndrome. This is mouse model work. And there's another drug, fingolimod, which is currently in clinical trials for multiple sclerosis. And there's some early studies in mice, Rett mice, to suggest it could be of benefit, and it works on BDNF to improve BDNF levels. So what about current clinical trials relating to Rett syndrome? Well, I guess the first thing to do is to just talk a little bit about what a clinical trial is, because you'll hear that bandied about, but it's actually quite a complex process. And the first thing is that clinical trials, the most important thing about a clinical trial is safety for the subjects involved in the clinical trial. So all clinical trials are registered or administered through ethics committees, either university or hospital ethics committees. And most clinical trials these days are also registered on various clinical trial sites and are carefully monitored in that way. When you look at the different types of clinical trials that we see, or the different phases of clinical trials, they have 4 phases to them. The first is phase 1, which is usually a drug has been identified as a potential therapeutic for a particular disorder. So the first thing to do is work out whether the drug is safe or toxic in any way. And so a phase 1 trial is usually done on individuals who don't have the disease, so they're normal healthy volunteers, to look at the safety and toxicity issues relating to the drug and how quickly it's cleared from the system and all that kind of stuff. Phase 2 trials are usually the early investigative studies done on a small number of individuals who have the disorder, the aim being to see how well the drug works and whether there are any toxic effects being seen in the patients. And phase 2 trials are most usually initially done in adults with the disorder, not children with the disorder. Phase 3 trials are if you've got pretty convincing evidence from the initial studies, you then move on to a larger study where you compare your new therapy with what's considered to be the gold standard. And right now there isn't a gold standard for Rett syndrome. So the alternative arm in the study for something like Rett syndrome would be some individuals not being on the therapy, so being on a placebo, the sugar water kind of preparation, and another half of the cohort being on the therapy that you want to test. These are usually larger studies, often need to be multi-centre trials, and this is where clinical trials start to become really expensive. And then the final is the phase 4, so if a drug's been seen to be effective and safe, goes onto the market and is starting to be used at in clinics, then the companies are obliged to collect ongoing data relating to the drug to make sure that we're not missing any issues relating to safety of the drug. So that gives you a sense of what clinical trials involve, and they take time. So let's turn to a couple of areas where clinical trials are currently underway. The first relates to IGF-1. So this is a growth factor. It has multiple different activities, but in particular relevant to Rett syndrome, it has positive effects on brain cells, on neuronal cells. It's important for a disorder that primarily affects brain. It crosses the blood-brain barrier, so there's a physical barrier that prevents drugs and chemicals from getting into the brain from the bloodstream. And so if you want to have a therapy for something like Rett syndrome, it has to be able to get across that blood-brain barrier. And it acts by stimulating the same pathways that BDNF works on. So again, BDNF seems to be an important player in all of this. And so in 2009, a group out of MIT in Boston did a study in Rett mice, giving them IGF-1 as it's an injectable treatment, and they saw very significant improvements in those mice, which ultimately led to a clinical trial starting at Boston Children's Hospital, initially started up by Omar Khawaja, who's since left Boston and is now working for Roche in Switzerland. And then the study was taken over by Walter Kaufman. He moved from Baltimore to Boston, and he's moved again more recently. So that's a clinical trial that's underway. Just to highlight how expensive these trials can be, it's costing around $2 million, and that doesn't include the cost of the drug. The company is actually giving the drug for free for the trial. But if this proves to be successful, then in terms of what IGF-1 costs off the shelf, you can see what it's going to cost an individual, about $40,000 a year to be on this therapy. So they're not cheap, and that creates its own hurdles when it comes to trying to get drugs like this in the Australian context. It is possible to get very expensive drugs for rare conditions through a program in Australia called the Life Saving Drugs Program, but there has to be very strong evidence before the federal government will even countenance making available the funds to do this kind of thing. There's also an oral preparation that's a synthetic version of IGF-1 that has been developed by a New Zealand-based company called Neuron, which also has offices in Melbourne, I heard last night. This, in fact, was initially developed and showed evidence of benefit in individuals with stroke and head injury. And There has been some work in the mouse model for Rett syndrome, and there's currently a clinical trial of this happening in the US as well, being led by Jeff Newell, I think. And so we're watching this space. Right now the clinical trial, as I understand it, has been going on in adults, but hopefully that will prove to be beneficial in adults, and then they will want to move on to clinical trials in children. And the Neuron people have certainly had discussions with us in the last couple of years, and we're waiting to hear back from them on whether they want to be ultimately proceeding with a paediatric trial. Desipramine is a drug that's been around for a while. It's an antidepressant, and it's been shown to improve the breathing abnormalities in the MECP2 mice. And so there's a French group that's currently, or has been recruiting patients into their trial. And this graph just shows the effect on the number of apneas that the Rett mice develop. So you can see that they have hundreds of apneas per hour, and the line stops at about halfway through because all of the mice died. That's a very severe disorder. Whereas the bottom line is mice that were treated with the desipramine, and you can see the number of apneas is dramatically reduced. So that's a clinical trial that's currently underway. Dextromethorphan, it's been around for a long time as well. It's a cough suppressant, and it too acts on a particular set of neurotransmitters. Neurotransmitters are these chemical messengers that help one brain cell communicate with the next brain cell. And there have been a number of abnormalities in neurotransmitters that have been described in Rett syndrome. And so people have thought, because there are lots of drugs relating to these types of chemicals, that these could be good targets to be attacking. And so dextromethorphan blocks a particular receptor, and currently Sakud Naidu at Johns Hopkins is running a clinical trial of dextromethorphan, and the particular thing she's interested in, what the effect is on seizures, the respiratory component, and the motor component of the disease. For a long time, people have wondered about whether abnormalities in mitochondria could be associated with Rett syndrome. Mitochondria are a little compartment within virtually every cell in the body. They look a bit like a bean, and they have about 1,000 different proteins in them that all have very important functions. And one of those functions is to convert the food that we eat into energy, into ATP. And that's the job of the respiratory chain, which is this bunch of red blobs here. And for a long time, or for some time, people wondered about whether mitochondrial disease could have been at the basis of Rett syndrome. And the reason for that was because there was quite a bit of overlap at a clinical level in Rett syndrome compared to patients with proven primary genetic defects of the mitochondrial respiratory chain. So that's the list of abnormalities that are shared in people with mitochondrial disorders and those with Rett syndrome. And in addition, there are a number of biochemical markers of mitochondrial disease that are abnormal in Rett syndrome individuals as well. So there's a chemical called lactic acid, which is sometimes elevated in the blood or in the brain. And prior to the MECP2 gene being identified, as part of the diagnostic odyssey that girls went through, or many girls went through, that often involved taking muscle biopsies to look for abnormalities in the mitochondrial respiratory chain. And in fact, some of them were shown to have abnormalities. So for some time, people wondered about whether it could be a mitochondrial disease. And then, of course, the MECP2 gene came along, which sort of changed the focus. But a number of us have been a bit of a dog at a bone at this, and there are, in fact, when you drill down, there are mitochondrial abnormalities in individuals proven to have, with Rett syndrome, proven to have MECP2 mutations. So some of the proteins that make the respiratory chain are reduced. Some of the genes that regulate the mitochondrial energy production pathways show abnormalities, and this is stuff that we've done and has also been recapitulated by others. And there's also evidence of oxidative stress in individuals. We did this work in the mouse model with Rett syndrome, but others have done it at a clinical level as well. So there's some evidence there to suggest that looking at improving mitochondrial function could potentially be of benefit in individuals with Rett syndrome. And indeed, there's a clinical trial currently underway funded by a company called Edison Pharmaceuticals, a drug called EPI 743, very sexy name, which was initially developed for a mitochondrial disorder called Leigh disease, a very specific mitochondrial disorder. And so this clinical trial specific for Rett syndrome is currently underway. People may have heard about fish oil and Rett syndrome. A group based in Siena in Italy, Claudio De Felici, studied this in a small cohort of girls with Rett syndrome. And again, it's based on the notion that hypoxia and mitochondrial dysfunction cause increased oxidative stress, and this can be measured in blood. So these are some of the things that they were able to measure and were shown to be abnormal in a cohort of girls with Rett syndrome. And polyunsaturated fatty acids, or fish oils, have an antioxidant effect, so they counteract the oxidative stress effects. And so they undertook a clinical trial, a pilot study in a small group of girls with Rett syndrome in so-called stage 1, so at a very early stage in the disease, and treated them for 6 months. Treated half for 6 months, and the other half weren't treated with a massive dose of fish oil on it every day. And they, in that 6-month study, showed improvements in some of the clinical features that we see in Rett syndrome. And so here, over the 6-month period, girls who didn't receive the fish oil showed no change in their clinical severity score, whereas girls that were on fish oil showed an improvement in the clinical severity score after 6 months. So it's a pilot study, only been done once. No one's replicated it or tried to replicate it at this stage. And there are still a lot of questions relating to this. What's the optimum dose? And they gave massive doses, not the usual 1 or 2 capsules, but really big doses, industrial strength doses of this. And if you look at fish oil, it's got 2 major components to it, EPI and DHA. What's the right ratio of those? How long do we need to be treating? Should we be treating before the girls become symptomatic? And really what's going on at the cellular level. So this is an area that I think warrants further study, because if fish oil can be applied and it's safe, then it could be a viable therapeutic. This pretty picture shows brain cells with what's called the microtubule network highlighted. So microtubules are structures within all cells, and in particular in brain cells, they're like tram tracks. Yeah, that'll be the right word to be using in Victoria. That can help move important things from one part of the cell to another part of the cell. And so this is the so-called microtubule network. And this is just a cartoon. This is the microtubule. Here's a little chap who's called a molecular motor, carrying his cargo along the microtubule network from one part of the cell to the other part of the cell. And it turns out that this is important in Rett syndrome. So what you can see here, we've taken brain cells from mice and been able to grow them in a dish and stain them with stain the mitochondria so that they show up as these white blobs. And all this movement you can see here is mitochondria being trafficking along the microtubule network. So this is what we see in a normal mouse, and this is what we see in a Rett mouse. And I think just by the eyeball you can see that there's a lot less action happening in the brain cells from the Rett mouse compared to the normal mouse. And we can quantitate that, and we can see that there's much less movement of mitochondria along the microtubule network, and there are other drugs as well, or other components. BDNF is packaged up into little granules, for instance, and is carried along the microtubule network as well. These microtubules are cylindrical structures, and they exist in a stable form and an unstable form. The stable form needs to have these acetyl groups attached to it, to the protein, the tubulin protein within, and there are enzymes that can put the acetyl groups on or take the acetyl groups off. And one of those enzymes that takes the acetyl groups off is called histone deacetylase 6, or HDAC6. And we've shown that in Rett syndrome mice and in cultured cells from males with MECP2 mutations, the histone deacetylase 6 levels are increased, which means there's less acetyl groups on the microtubules, which makes the microtubules unstable, they fall apart. And so that led us to postulate that if we could find drugs that could switch off HDAC6, that might stabilize the microtubules and might improve overall function. And so we've done that in cultured cells. So these are skin cell lines from two boys with different MECP2 mutations, and it's a bit complicated. I won't walk you through it except to say that when we used an HDAC6 inhibitor called tubistatin A, we could actually quantitate stabilization of the microtubule network in these skin cells. So showing that in skin cells is one thing, but what about in real life? And so we've been working with a medicinal chemist in the US, a guy called Alan Kozakowski, who essentially invented this class of drugs. And he tweaked the drug to be able to have better penetration into the brain. And so we've done some, this is very preliminary, but we've done some preliminary work in the mouse models. So this is a normal mouse doing the so-called open field test. And you can see he's very inquisitive, walks around the perimeter rather than in the middle of the field, because if he's in the middle of the field, the owl will come down and get him. And you can see he's inquisitive and so on. So that's what a normal mouse would do. This next one is a male Rett mouse at 10 weeks who's been treated with a placebo, so sugar water. And you can see he's really not looking very well at all. He's hardly moving at all. You probably can't see it very well, but he too has a tremor and he's got breathing abnormalities as well. So that was a male mouse treated with a placebo. And then the next video is another male mouse, Rett mouse, treated with this the drug for a period of 4 weeks, having been started at 6 weeks. And you can see he's looking a whole lot better. He's still not walking normally, but he's looking much better, is inquisitive and so on. So this, we're pretty excited about this. There's obviously still a lot of work to do. The money really is going to be in what does this class of drugs do in the female mice, because that's really what's going to be most important for most individuals with Rett syndrome. Unfortunately our money's run out, and we're hoping to try and get some funds from other sources so we can go on and do that experimental work. But just to summarise what I've been talking about, firstly, there's good evidence that abnormalities in function of mitochondria may be important in contributing to the disease phenotype in Rett syndrome, and so that could be potentially tractable as a potential therapeutic. All of this really boils down to our understanding of the biology of Rett syndrome has improved so dramatically in the last 10 years or 15 years that we can now start to rationally tease out potential therapies, including drugs that might restore microtubule dynamics, drugs that can increase BDNF levels or focus on the targets of BDNF, the so-called nonsense read-through drugs for the third of individuals with Rett syndrome, and there are others coming down the pipeline. So more clinical trials are going to be happening. I think it's fair to say that there is light at the end of the tunnel. I would hope that in the next 5 years or so we're really going to see some very, very tractable therapeutics for Rett syndrome. And I'd just like to finish off by acknowledging people who shared some of their unpublished data with me that allowed me to give this presentation. And I really need to pay homage to a bunch of really talented and enthusiastic PhD and master's students that I've had along the way. Some of whom were silly enough to stay on with me and do postdocs, but who were, also very enthusiastic. And most recently, I have to target Wendy here, who is a real little dynamo. She's about this tall, but absolutely is totally immersed in Rett syndrome research. And I'm hoping I can find some money to keep her working with me in the lab so that we can progress this work. And of course, senior research colleagues. You've heard from Helen. Helen and I guess have published about 30 odd papers together, yeah, more. But I've had other colleagues in Sydney, in WA, in Canada, and in Wales, who I've been very actively involved with in terms of my research. And finally, our various funding sources, and in particular, I want to pay tribute to Janelle Lillis, who received an award yesterday. Janelle and her fundraising activities, and she really motivated Rotary as well. Without her initial and ongoing support over a number of years, we wouldn't have been able to do at least 50% of the stuff that we've been able to do. So without Janelle, we wouldn't have even got on the map. So I really want to thank Janelle for all her support over the years. And on that note, I'll stop, and I'd be happy to take any questions if anybody has any.
We'll have to move on. But just in case you do win the auction today, celebrate for that. Thank you very much.